Now, when you look at this table, whichever side it is on my head, you'll see that well, lots of negatives and lots of positives in this table when it comes to prokaryotes versus eukaryotes. Means that while we do have some things in common, we have DNA, we have ribosomes, we have metabolism, things like that, prokaryotes, eukaryotes are quite different cells. We have internal membranes, we have organelles, we have multiple chromosomes, we have our genomes are much, much larger. So, because of that, eukaryotic cells are able to be bigger, are able to do more, a lot more going on. But please don't think that prokaryotic cells are rudimentary, that are so elementary they can't do much. We're going to see throughout the rest of this semester, there's quite a bit going on with bacterial cells. Now, the question always is, where did we get these internal organelles? How did we become eukaryotic? We don't know. I could do an experiment to figure it out and all that stuff. It'll take me a few 100 million years, because that's basically how long we thought it took. When we look at modern day prokaryote and eukaryotic organisms, we see that there is almost steps. Things started here, things were maintained through the lineage of this microorganism for the past 3 billion years. Whereas other organisms, they evolved, they advanced, more abilities, more adaptations. So you can see that there is almost not quite a path, but it gives us an idea of what had to have occurred. If you look at the figure to, the left of, my talking head, you will see that billions and billions of years ago, everything started out as that protocell, that first cell. We don't know much about it. Again, had a membrane, had some DNA or RNA, had some enzymes to do a metabolism. Over billions of years, this protocell group split off into different types, as I discussed before. One of the groups, one of the camps, which would become what we now know as eukaryotes, that membrane pushed in, started to wrap around stuff. Now, it wasn't quick like that it happened. No, this is over, again, millions of years. So, what we think happened is that as it pushed in, wrapped around, protected the chromosomes, those cells did a little bit better. Less UV damage, less going on to damage their chromosomes. They did better. Now, the other cells that didn't do this, they suffered. They struggled. Well, you fast forward a few million years, well, instead of just the plasma membrane now wrapping around some of the chromosomes, you have true nuclear envelope. So those chromosomes are completely protected. They do a little bit better. They do a little bit better. They outcompete just a little bit better more. A little bit more, a little better. That's evolution. Early eukaryotes, early archaea are around each other, or early bacteria. We're not quite sure which. The early bacteria, we'll go with those first. Early bacteria around those early eukaryotes. Bacterial waste, because it makes too much of stuff, now becomes food or things for the early eukaryote to use. Early eukaryotes waste, because it makes more than it can use, now becomes the bacterial's food stuff. They do better when they're in close proximity than further apart. Fast forward a few million years. Instead of being around each other, they're now attached to each other. That way they can directly back and forth. Those cells that are attached do better than the ones that are just floating around each other. They outcompete just a little bit better. Evolution. Fast forward a few million years. Instead of being attached on the outside, it's now on the inside. It's still a separate living bacteria, but they are now protected. They now have a constant supply of food sources. They make excess things, ATP being one of them. They pump it out. It's now there for that host cell to use. They outcompete the ones that are attached, which are outcompeting the ones that are floating around each other. Evolution. Fast forward a few million years, now instead of being a bacteria inside of a cell, it's lost all of its ability to be in a bacteria. It's now the mitochondria. How do we know this? Well, think back to concepts. Intracellular molecular. The mitochondria has dual membranes, has its own genome, its own ribosomes. Wow, that's pretty much sounds like a bacteria. Dual membranes for the gram-negatives, as we talked about last module. It just lost its thin cell wall. Same thing happened with archaea, or some of the photosynthetic bacteria, which now became our chloroplasts.